I would like to introduce Liliana Panetti. She is the COO of our organization, and Liliana will introduce our featured speaker today.
Thank you very much, Dr. Warren. I'm the Chief Operating Officer for the Partnership for Maternal and Child Health of Northern New Jersey, and I have the great honor and privilege to introduce our guest speaker, Dr. Paul Offit, an internationally recognized expert in the fields of virology and immunology and the world-renowned vaccine advocate. Dr. Offit is the Director of Vaccine Education Center at the Children's Hospital of Philadelphia, as well as the Maurice Hilleman Professor of Vaccinology and the Professor of Pediatrics at the School of Medicine at the University of Pennsylvania. He is a recipient of many honors and awards, including the G. Edmund Bradley Prize for Excellence in Pediatrics from the University of Maryland Medical School, the Young Investigators Award in Vaccine Development from the Infectious Disease Society of America, and the Research Career Development Award from the National Institute of Health. Dr. Offit has published more than 160 papers in medical and scientific journals in the areas of rotavirus-specific immune responses and vaccine safety. He's also the co-inventor of the rotavirus vaccine, RotaTeq, recommended for universal use in infants by the CDC in 2006 and by the WHO in 2013. Dr. Offit is also the author of 9 medical narratives, including Vaccinated, Deadly Choices, Bad Advice, and the most recent book, Overkill, When Modern Medicine Goes Too Far, which was just published in April 2020. In Overkill, Dr. Offit takes on 15 common medical beliefs that are held by the general population and medical professionals, but not supported by scientific evidence. I just finished reading Overkill and I highly recommend it. It is clear, simple, and informative, a book aiming to help all of us to become better advocates for our own health. Today, Dr. Offit will present the path to SARS-CoV-2 vaccine, focusing on strategies, challenges, and how to manage expectations following the release of the vaccine for COVID-19. CME and CEU credits are being offered for this program. Many thanks to our Board of Trustee Chair, Dr. Lamacchia, and the staff at St. Joseph University Medical Center for providing CMEs for today's program. 1 hour following the program, attendees will receive a link to an online evaluation. Please complete it, indicating which kind of credit you prefer. Dr. Offit, we are very excited about your presentation. Thank you for joining us today. Now go to you.
Thank you, Liliana. Thank you for that kind introduction. So I'm looking at myself here. Let me just close this part out so I can see. Okay, good. People can see you. You've hidden your webcam, but still start showing. Okay, I'm just great. We'll go here. Okay, hopefully I can move this thing out so I can actually see my screen. Okay, good. So what I thought I would talk about today is really the only way to end this pandemic completely is with a widely used vaccine against SARS-CoV-2 virus. I think Tony Fauci said it best. The only way we can feel comfortable having large-scale sporting events is with a vaccine. So I think there will be a vaccine. I think that there will be a safe and effective vaccine. The only thing I worry about, and we'll get to this in a little while, is that it goes through the process that all vaccines normally go through. Given that we're making this vaccine faster than any vaccine that's ever been made before. The fastest vaccine made before this was the mumps vaccine. When Maurice Hilleman isolated the virus from his daughter, Geraldine Hilleman, in 1963, that was a commercial product in 1967. That was a 4-year development effort, and that was the fastest vaccine to date. This is going to be faster than that. This is going to be probably about a 1.5-year cycle, because I really do think we can have a vaccine by the middle of next year. Okay, so what I'm going to do here is review the basic strategies in making a vaccine, explain the challenges in making this particular vaccine, and then discuss how to manage expectations following the release of a SARS-CoV-2 vaccine. And probably the better way to say the last part is vaccines. I think we're going to have more than one vaccine in this country. I think we'll have a few vaccines in this country, and there'll be far more than that in the rest of the world. I have nothing to declare other than I am a diehard Philadelphia Eagles fan, which makes me incapable of actually evaluating how well this team will do. Okay, so successful completion of this program is to say to receive continuing educational hours for this presentation, you must attend the entire webinar and complete an online evaluation. A certificate of attendance will be sent electronically within 1 week of the program. This program will be available on the partnership's YouTube channel until June 9th, 2022. All right, so let's start with the good news. The good news is that there has never been more expertise, more money, or frankly, more of a will to make a vaccine in human history than this vaccine. There are more than 120 investigational new drug licenses that have been submitted to the Food and Drug Administration. More than 70 companies across the globe are interested. I mean, typically, when you make a vaccine, 1, 2, at most 3 companies are interested in making that vaccine. There's never been a greater level of interest for obvious reasons than for this vaccine. BARDA, which is part of Health and Human Services and stands for the Biomedical Advanced Research and Development Authority, has already given actually more than $2.5 billion to 5 different companies. The virus is relatively stable. This is not influenza, although it's a single-stranded RNA virus like influenza. This virus does not mutate so much from 1 year to the next that natural infection or immunization the previous year does not protect you the following year. So I think the vaccine will provide likely protection against moderate to severe disease for several years, not decades, but I think for several years. And BARDA, by the way, is one of just many groups that are interested in paying money to move this vaccine forward. World Health Organization gives money to this. The Bill and Melinda Gates Foundation has given money to this, and as well as other private and public institutions. The other thing is the protein you're interested in. You're interested in that spike protein, which emanates from the surface of the virus. That's the cell attachment protein. I mean, that's the protein that allows the virus to attach to and enter cells. If you can prevent the virus from binding to cells, i.e. by making antibodies to that spike protein, you can prevent the virus from infecting cells, or saying another way, you can prevent the virus from causing disease. So the approach that's being used to make this vaccine includes the approaches that have been used to make every vaccine that we've ever made before, as well as some novel ones. So I'll go through that. So one approach is to take the virus and kill it, which is the way that we make the inactivated polio virus vaccine, the hepatitis A vaccine, and the rabies vaccine. Probably the group that's the farthest along on this particular approach is China. There's a China group that's fairly far along here, and it'll be interesting to see how that plays out. Their data look excellent, and I know that the administration, our administration, the administration in the United States, has said that they will accept no vaccine that's made in China. But again, if this is the best vaccine, the safest vaccine, the most effective vaccine, we may have to reconsider that original statement. The other approach is the live attenuated viral vaccine. So what you do there is you take the virus, you weaken it in the laboratory so that it can reproduce itself to some extent, but not so much that it can cause disease, but still reproduce itself so that it can induce an immune response, which is protective. That's the way that we make the measles vaccine, the mumps vaccine, the rubella or German measles vaccine, the chickenpox vaccine, and one of the rotavirus vaccines. The third approach, and these are all approaches for which we have existing platforms, is to take the protein you're interested in, which is that spike protein, and then give just that protein. Usually this is done using a recombinant DNA system, and this is the way that we make the hepatitis B vaccine and the human papillomavirus vaccine. In those cases, what you do is you take a yeast plasmid, you insert into that yeast plasmid the gene that codes for the protein you're interested in, whether it's the HPV surface protein, which is called the L1 protein, or the hepatitis B protein, which is called hepatitis B surface antigen, and then that's what you give. You give about 20 micrograms or more of that viral protein. The fourth possibility is the so-called vectored vaccine. So here what you do is you take, say, if you take in the case of dengue, you take a yellow fever vaccine virus, which doesn't reproduce itself very well, and then you clone into that the gene that codes for the SARS-CoV-2 surface protein, the spike protein, the S protein. That's a similar approach that's used for the current Ebola virus vaccine, which is done by Merck and in collaboration with the National Institutes of Health, which is you take a vesicular stomatitis virus vaccine, which is not a human virus, and so it reproduces itself very poorly in people. And then in the case of Ebola, you clone in the genocodes for the Ebola surface proteins. So you see the similarities in all these approaches. The last three are not platforms which currently exist. So that the examples would be take a replication defective adenovirus. So for example, the Johnson & Johnson has a replication defective adenovirus 29. Now what I mean by replication defective is the virus cannot reproduce itself. However, it can make proteins. In the case here, the protein we're interested in, which is the spike protein. This is what's being done by Johnson & Johnson, who uses an adenovirus 29 vector. It's done by the group in the United Kingdom at the Jenner Institute at Oxford, where they use a simian replication defective virus. And it's also being done by a group in China, which uses an adenovirus 5, which is a little more common in the circulation, so may perform less well than the adenovirus 29, which is not as common in the circulation. And all those existing platforms are being pursued. Those non-existing platforms are being pursued. The next platform, and this is probably what you've read the most about, which is messenger RNA. So what you do here is you inject the messenger RNA that, again, codes for that surface protein, the spike protein. It's taken up by dendritic cells or macrophages, both of which are antigen-presenting cells, and then those antigen-presenting cells then make that protein. So now you're not giving the protein, as in the case for hepatitis B or human papillomavirus. Rather, what you're doing is you're giving the genetic material that allows the person's body to make that protein. And then finally, DNA, which is essentially the same idea, it's just one step earlier. You give a DNA that then codes for that spike protein. The DNA is then transcribed to messenger RNA and then translated to that protein in your body. So again, you're making the protein, which is true of all of the last three vaccines. Now, I think the reason that you hear the most about the DNA vaccine, which is being pursued by Inovio, which is a Plymouth Meeting, Pennsylvania company, the messenger RNA vaccine, which is being produced by Moderna, which is a Cambridge, Massachusetts company, or Pfizer, which is also producing messenger RNA. And then the replication defective adenoviruses, the reason you hear the most about these three is they're the fastest to make. They're the easiest to make. All you have to do is take that genetic material and plug it into your system. It's much harder to make a whole killed virus. It's infinitely harder to make a live attenuated viral vaccine, and to some extent a purified viral protein vaccine, which, by the way, is being pursued by Novavax. So I think the reason you hear the most about these last three is not because they're necessarily safer, not because they're necessarily better, meaning more effective. It's just because they're the easiest and fastest to make, and obviously there's a tremendous interest in trying to make these vaccines as quickly as possible, because we're dealing with a virus that's killing between 1,000 and 2,000 people a day in the United States and far more in the world. Now, oops, sorry. Okay, so as I said earlier, or maybe I didn't say earlier, but typically, if you're making a vaccine, it usually takes about 15 to 20 years. I mean, I was fortunate enough, as Liliana mentioned, to be part of a team at Children's Hospital of Philadelphia that created the rotavirus vaccine, RotaTeq. That took 26 years. That's not that unusual. So if we're making a vaccine in 1.5 years, why is it we're able to do this so much faster for this vaccine than for previous vaccines? Well, so let's go through how you typically would develop a vaccine. Typically what you do is you develop an animal model. So you inoculate an animal, in this case with SARS-CoV-2, and you hope that it develops signs and symptoms consistent with that virus, so that it mimics the human infection. Then you try out your different ideas. You give whole killed virus, you give a weakened virus, you give just a purified protein, and then you see whether that animal model, in that animal model, you can actually show that your vaccine protects. These are so-called proof of concept studies. You don't have to do them. It's nice to do them, primarily because you can then, I think more importantly, look at what part of the immune response is associated with protection against disease. Because obviously you can do far more with mice than you can do with people in terms of evaluating an immune response, especially with an inbred animal. So, so that has generally been either skipped or truncated with this vaccine. Then what you do is you do dose ranging studies. So now you think you've got the right idea for how to make a vaccine. So you try a variety of doses to make sure you're not giving too much vaccine virus and not giving too little. Usually those studies involve hundreds and occasionally thousands of people. For this vaccine, I haven't seen really any dose ranging studies that are much more than low hundreds. So again, it's a little more truncated with this vaccine than it's been with other vaccines. Then you do progressively larger safety and immunogenicity studies in hundreds to thousands of people. As you move through phase 1 and phase 2, what you're doing is you're learning about whether your vaccine is safe as you move to more people. And most importantly, you're learning whether it's consistently immunogenic, meaning not occasionally immunogenic or largely immunogenic, that it's consistently induces the immune response you want it to induce that you think is associated with effectiveness. But I can't underscore this enough. You don't know that. You don't know whether or not your vaccine is effective until you do what is a phase 3 trial. That's a large, prospective, placebo-controlled trial involving tens of thousands of people. So for example, our vaccine was tested in more than 70,000 infants, meaning 35,000 got vaccine, 35,000 got placebo, and that was done over a 4-year period. RotaTeq, another rotavirus vaccine, was tested in 60,000 babies before it was licensed by the Food and Drug Administration. 30,000 got vaccine, 30,000 got placebo, and that was also done over several years. The human papillomavirus vaccine was tested in phase 3 trials in about 30,000 people, primarily women, over a period of 7 years. The conjugate pneumococcal vaccine, the first one, PCV7, not PCV13, was tested in about 35,000 people, again, prospectively in a placebo-controlled study, because the only way you can know whether your vaccine is effective is to do these studies. Immunogenicity is not enough, as we've learned again and again and again. There is no immunological correlate for protection against rotavirus vaccine with RotaTeq. There's not. I mean, we had children who got rotavirus vaccine who got sick with rotavirus. We have children, obviously most of them, who got rotavirus vaccine that never got sick. We had serum on every one of those children, yet we were never able to find a clear immunological correlate of protection using serum. I think if we had biopsied the intestines of all of those children, which obviously no parent in the world is ever going to agree to, if we'd biopsied the intestines of those children, I think we would have found that probably the immunological correlate was memory B and T cells in the lamina propria of intestines, because that's what it was in mice. There also wasn't a clear serum correlate in mice either. So we found an immunological correlate with mice because we can do things to mice that we can't do to people. And so I think realize probably half the vaccines on the market right now don't have a clear immunological correlate of protection. So if you hear someone say, look, this vaccine appears to be safe in a few thousand people, and we have consistent immunogenicity, don't believe that immunogenicity necessarily correlates with effectiveness, because you don't know that until you do a phase 3 trial. Okay, so let's just sort of to redo what I just said. So as I said in the previous slide, there have been for this vaccine no or limited animal model studies. There are very small dose ranging studies. Now the phase 3 trials that are planned here, and I'm actually on a group that was put together by Francis Collins, who's head of the National Institutes of Health, to try and determine what we want to see from basically these warp speed vaccines. And I'll tell you what warp speed is in a second. What we want to see is a prospective placebo-controlled efficacy trial that involves at least 20,000 vaccinees and 10,000 placebo recipients. Now, warp speed is another thing that has enabled us to go faster here. Typically, when a company makes a vaccine, they do a phase 3 trial, they find it safe and effective, and then they mass produce it. Warp speed is different. What warp speed is the companies are making the vaccine, mass producing the vaccine at risk, meaning they don't know whether their vaccine is safe. They don't know whether it's effective because phase 3 trials haven't been done yet they're mass producing it, assuming that the vaccine will be safe and effective. Now, what's also happened here is some companies are not going to be doing this, are going to be sort of doing stuff away from the NIH recommendation here and do much smaller phase 3 studies. And remember, these are not going to be FDA licensed products. Typically, when a vaccine is made, it goes through the FDA, and the FDA has something called the Vaccine Related Biological Product Approval Committee, or VRBPAC committee. That's the FDA's Vaccine Advisory Committee. I sit on that committee, as well as a number of people who are experts in this field. And we're not going to see these viruses. We're not going to see these vaccines. We're not. This is going to be these are not going to be licensed products. They're going to be approved products under emergency use authorization. I mean, basically, you saw what happened with hydroxychloroquine. That was another example of that, which is that the FDA has never licensed hydroxychloroquine for the use in SARS-CoV-2 because it's never been shown to work to treat people with SARS-CoV-2. And in fact, 3 studies have now been done, prospective placebo-controlled studies, after it was already allowed to be used, showing that the hydroxychloroquine didn't work. And more importantly, 10% of people who participated in those trials had to drop out because they developed cardiac toxicities, primarily arrhythmias. And so I don't think that was the FDA at its best. I think that the administration very much wanted there to be a magical drug that treated this. It was touted by the administration. And we've learned now that it didn't work. And I think one thing that the president said is he said, it can't hurt. Well, that's not true. any drug can hurt. I think people assume that when you're sick, that you can't really hurt someone who's sick, but you can hurt someone who's sick. I mean, if someone's sick, there's 3 possible outcomes. Either they can get better, they can stay the same, or they can do worse. If the product doesn't make you better, then it can do only one of 2 things: either nothing or make you worse, which is what we've learned with hydroxychloroquine. The things that worries me the most on this, and I wrote an op-ed piece with Dr. Ezekiel Emanuel at Penn that was published in the New York Times on Monday. What's today? Wednesday, Monday, 2 days ago. What I worry about is what I will call the October surprise. There's an election coming up. It's going to occur at the beginning of November, and I think there is an intense desire to have a vaccine before that election. I think Dr. Fauci has said it. He said, we hope to have this vaccine ready by October. Deborah Birx has said it, and Donald Trump has said it. He's made this clear. He wants to have a vaccine by October. And what worries me in this is I don't think there's any way we can do a 20,000, 10,000 placebo-controlled study, vaccine placebo-controlled study by October. We're just starting in July. That's 30,000 people who you have to recruit, and you have to be lucky that you're recruiting those people in areas where there are outbreaks. So you have to pick the right areas to do that, because you have to have enough people in your placebo group who get sick with this virus so that you can actually make a statement about efficacy. And I worry that what they're going to do is just say, look, we have great immunogenicity. The vaccine appears to be safe in a few thousand people. Let's put it out there. Let's take 1 or 2 or 3 of these BARDA-funded vaccines, all of which are in warp speed, and put it out there. And just to give you an example, this warp speed program is not unprecedented, or said another way, it has a precedent, which was polio. I don't know if any of you remember this, but because I'm probably the oldest person on this call, but I remember polio. I mean, I was a little boy when polio was a scourge, and polio in the mid-1950s caused 30,000 people to be paralyzed, as many as 30,000 be either paralyzed for the rest of their life or an in and iron lung, often for the rest of their life, and it caused 1,500 children to die in this country every year. Jonas Salk, when he was doing his trials of an inactivated polio vaccine, tested it in about 700 children in the Pittsburgh area, finding that it appeared to be safe and that it was consistently immunogenic, consistently. But we waited for a phase 3 trial. Now, during that phase 3 trial, which was, I think, probably the largest medical trial in history, we gave that vaccine to 420,000 children, first and second graders in this country, and 200,000 children received placebo. 1.2 million children were not given anything. They were just observed uninoculated control. So that was a 1.8 million child study. I think that study, if it was done today, would probably cost $5 or $6 billion. It was funded by the National Foundation for National Foundation for Infantile Paralysis, otherwise known as the March of Dimes. They funded that. In other words, it was funded by a private philanthropic organization. At the same time that they did that placebo-controlled study, we had our own warp speed. 5 companies made that vaccine at risk. Now, it was no risk to them because the National Foundation for Infantile Paralysis, or March of Dimes, paid them to do it. So if the vaccine was shown not to be safe or not to be effective, they wouldn't have been out any money, and we would have just had to throw all that vaccine away. But it was safe and effective, and so we launched the polio program. And people will say now, and I just want to again say I think this is a bad argument. People will say, look, we need to skip steps because this is a scourge, because this is killing 1,000 to 2,000 people a year in the United States. I'm sorry, a day in the United States. But polio was no less terrifying and no less paralyzing. I mean, we certainly were paralyzed by that virus, and this was killing children. I mean, this virus was killing and paralyzing children every day. and over the summer months. And so I would argue that virus was no less terrifying than this virus. So if we could wait then, we can wait now. So I think there will be a vaccine or vaccines. I think they will be released in this country. I predict that they'll be released by, if we do it the right way, if we do placebo-controlled phase 3 trials, I think we will have a vaccine by the middle of next year. I just want to make sure that we do these phase 3 trials, hence the op-ed that Zeke Emanuel and I wrote. So I think once these vaccines are released, I think we're going to have to do a very good job at managing expectations, because this can go one of two ways. Either vaccines will be the hero of this story, they'll protect ourselves, they'll protect the adults and children who are infected by this virus, and we'll get back to our way of living. And I think we can do that with roughly an 80% immunization rate. But I think regarding safety, we have to make sure that when you test something in 20,000 people, that's not the same thing as testing it in 20 million people. So there may be safety issues regarding this vaccine that we're only going to find out post-licensure. Now, that's okay. I mean, that's why we have the vaccine safety data link. We have systems in place that will allow us to determine whether or not there are any rare side effects. You can rule out common side effects pre-licensure. If you do a phase 3 trial, as I mentioned before, you can rule out relatively uncommon effects pre-licensure, but you're never going to rule out rare adverse events pre-licensure because you just can't test it in enough people. Maurice Hilleman, who I consider to be the father of modern vaccines, he's made 9 of the, either did a primary research or development on 9 of the 14 vaccines that we currently give to infants and young children. He said it best. He said, quote, "I never breathe a sigh of relief until the first 3 million doses are out there." And that's always true, and you're not going to do that kind of study pre-licensure. So, but the good news is we do have systems in place to detect those rare adverse events, primarily the vaccine safety data link, and to a lesser extent, the vaccine adverse events reporting system. Regarding efficacy, we're certainly not going to know duration of protection when these vaccines first roll out. I think given that this is a respiratory virus where viremia is not an important part of pathogenesis, we're not going to get sterilizing immunity like we get with measles, where you're protected against all forms of disease, mild, moderate to severe, and frankly protected against reinfection associated with exposure. That's not going to be this vaccine. I think what we can likely expect from this vaccine or these vaccines is that we'll get protection against moderate to severe disease, which is to say we'll be able to keep people out of the hospital and we'll be able to keep them from dying. I think that's what you can expect from this vaccine. But again, you have to manage that expectation. I think if these vaccines roll out then and people find, wait, I'm still getting mild disease, I still might be shedding virus, we have to make sure people understand what to expect and what not to expect from these vaccines before they roll out. So again, I think much is at stake here. As I said earlier, this will play out one of two ways. Either it'll play out the way the end of the movie Contagion played out, which is that you see people lining up for the vaccine because it's been shown to be safe and effective, and vaccines are seen as the hero of this movie. Or it could play out the other way, which is because we don't do what we need to do correctly, which is to say a large prospective placebo-controlled phase 3 trial, we may find out that the vaccine is far less effective than we might have guessed based on solely immunogenicity studies, and that it has efficacy, say, less than 50%, 40%, 30%. That will be disappointing. And I think it's a fragile vaccine confidence in this country right now. And I'm not talking about the anti-vaccine people. They don't like what we do no matter how safe or effective vaccines are. They don't trust us. They're conspiracy theorists. I'm not talking about them. They have no confidence in us. And they are a very small percentage of the population. They're the mouse that roared. They have the ability to get their voice out there primarily on social media, because I think, frankly, mainstream media has largely turned their back on anti-vaccine activists. I'm talking about the rest of us. I'm talking about those parents who are perfectly willing to give their children vaccines to prevent 14 different diseases in the first few years of life that can mean as many as 27 inoculations during that period of time. It can mean as many as 5 shots at one time to prevent diseases most of them have never seen, using biological fluids that most of them don't understand. That's confidence. We have their confidence. We have their trust. And that's what worries me. If we put a vaccine out there that we haven't thoroughly tested to be safe, and at least as safe as it can be, and at least thoroughly tested it to be effective, at least as effective as we can know. I mean, to do a phase 3 trial is to mitigate risk. It's to make us feel better that we have an effective vaccine and that we have a safe vaccine, but it doesn't prove it. I mean, the proof invariably comes post-licensure when we do phase 4 post-marketing studies, and we do the kinds of studies that are done with the vaccine safety data link by academics. That'll be the final proof, but at least it lessens the risk we have a severe side effect problem, and it lessens the risk that we're underestimating effectiveness. So that's all I have to say. I will take your questions and happily, hopefully that made sense. Thank you.
Thank you very much, Dr. Offit. This was great, very informative. I have already a few questions from the audience, and I would like to encourage everyone to type questions in the chat box, in the question box for us, and I'm going to go through them. The first question we have, it's does the virus, this virus, the SARS-CoV-2, survive the acid environment of the stomach and then infect the cells of the intestine? That's a question from Dr. Slater. Thank you.
That's a great question. Certainly, we're seeing in the children, for example, that have SARS-CoV-2 infection that seems to be associated with this multi-organ inflammatory disease of children, that some of those children present as if they have cholera. I mean, it's this massive diarrhea. Now, technically, this is this is an envelope virus, so it really shouldn't survive the acids and hydrolases and proteases that are in the stomach. It shouldn't. Now, so the question is then, in these kids who have diarrhea, and some also adults who are infected with this virus can also develop diarrhea, is that because virus is replicating in intestinal epithelial cells or not? And so we really need studies to answer that question. Those studies are best done post-mortem, actually, but so I don't know. I mean, you look at the kids, for example, who have multi-system disease, which is basically a vasculitis. I mean, some of those children have died. A handful of those children have died. It would be interesting, assuming the parents allow autopsy, to look in those endothelial cells where there is a vasculitis and see whether or not there's virus there. To look in the intestines to see whether there's virus there, because the other option is that it's all cytokine and chemokine mediated. It's like flu, for example. Influenza virus can cause rhabdomyolysis, severe rhabdomyolysis, which can cause kidney failure. But if you look in those muscle cells where rhabdomyolysis is occurring, you can't find infectious virus, because influenza, like this virus, near as I can tell, really doesn't cause viremia. So you wouldn't expect it to travel to distant sites and cause those problems. So I think this all may be chemokine, cytokine mediated, but again, we wait to figure this out. And this is another thing, by the way, not to completely depress you, but this is a bat coronavirus that has just made its debut in the human population. There's a lot we don't know. There's already been a couple of surprises. I mean, one surprise is that it's more contagious than we'd originally imagined, more contagious than influenza. The second surprise is it causes this apparent post-infectious multi-system, multi-organ vasculitis disease, which had never been seen before with human coronaviruses. And the third thing, and this is a sort of an insider's baseball kind of thing, is that there are some people who are infected with this virus, develop clear signs and symptoms, who never develop neutralizing antibodies. That's amazing. I mean, the spike protein on the surface of this virus, if they didn't do the antibodies, they should be neutralizing antibodies, because they're going to prevent virus cell binding at the so-called receptor binding domain. Yet there are people who never develop a neutralizing antibody response who are clearly infected, which makes you wonder whether there may be some immunosuppressive component with proteins other than that surface proteins. I mean, that is another thing being looked at. So be humble. As you move forward with these vaccines, be humble, which is to say there's much we don't know right now. And I can assure you, as we move forward on these vaccines, we're going to learn things, some of which we may not want to know. And I get a little tired of this kind of science by press release, where groups like Moderna or J&J, the AstraZeneca, who has affiliated themselves now with the Jenner Group in the UK, which has the replication defective simian adenovirus. Stop doing this. Stop talking about how wonderful your vaccine is going to be. You don't know that yet, so just shut up, do the phase 3 trial, and then you can start talking.
Yeah. Thank you. One question I have ready for you is, what are your suggestions for reassuring patients that it's safe and necessary to receive SARS-CoV-2 vaccine when they have safety concerns regarding receiving a new vaccine?
Well, they should have safety concerns. I think we all should be vaccine skeptics. I mean, I'm on the FDA's Vaccine Advisory Committee. I consider myself a vaccine skeptic. I want to see the data that reassures me that these vaccines are going to be safe and effective. So I think, again, assuming the phase 3 trials are done the way they should be done, we'll have more information about what to tell parents about what we know and what we don't know about this vaccine. So it's always a matter of relative risk. I mean, everything we do in medicine is a matter of relative risk. We want to do the thing that puts you in the best, safest position possible, but everything's a risk. I mean, taking a vaccine is a risk. I would argue that that risk is much lower than a choice not to get a vaccine, where you're taking a different and more serious risk, a greater risk. So that's always true here. So I just think we have to be honest with people about what we know and what we don't know going into this. But you do have a virus that's killing about 1,000 people a day here in the U.S. So that's the risk of not getting a vaccine. And so now we need to know what the risk of getting a vaccine is, and we'll learn that, assuming we do this the right way. Assuming that we do this the way every vaccine has ever been made in this country since the 1950s. Don't veer off that. And it really does worry me. That's why I wrote that op-ed piece, which did not lessen my hate mail from Trump supporters, by the way.
And a follow-up question to that. At what point would you feel comfortable receiving a SARS-CoV-2 vaccine yourself?
I'll look at the phase 3 trials, and I will evaluate those trials based on data, and then if I'm comfortable that the vaccine at least is as safe as we can make it, knowing we're only doing, say, 20,000 people, and we know that it's, say, effective at a 70% level, 80% level, I'll get the vaccine.
I have a few questions regarding vaccines for children and pregnant women. So my question is, when do you anticipate a vaccine being available for use for children and pregnant women?
Right, so this is always true. When placebo-controlled trials are done, phase 3 trials are done, pregnant women are excluded. Or let me rephrase that. Pregnant, we try and exclude pregnant women. We never do, not surprising, because not everybody who is pregnant necessarily knows they're pregnant early on. So so there's always pregnant women in pre-licensure trials. I don't think children are going to be part of these pre-licensure trials. I can tell you the way that the NIH active group has designed these trials for these BARDA vaccines, the so-called warp speed vaccines, children are not going to be included initially. The children will be tested after the vaccine is already approved and out there. And the main reason for that is that children really aren't likely to be killed by this virus, so they're not a priority group. And I think when we finally release the vaccine, I think that you'll probably see, hopefully the Centers for Disease Control and Prevention, CDC, will have sort of a phase in for this vaccine. So for example, phase 1 would be healthcare workers, people who work in meatpacking plants, grocery stores, pharmacies, mass transit, that they will be the priority group, and then we'll work our way down. But I can tell you that children will very quickly be studied once there is or are licensed vaccines, or I should say approved vaccines.
I have a question from one of our staff about the plan to promote this vaccine to the community, specifically in the Black community, where trust of medical profession in general vaccine, it's, low.
Yeah, first of all, I think trust in this vaccine is not high, period. I mean, I've talked to a lot of people, nurses, the people who are just on the front lines, and a lot of people are very hesitant about getting this vaccine. And I think the reason for that, and I can't tell whether it's more likely, say, an African-American community or communities of color would be more likely to reject it. Certainly the African-American community has reasons to be suspicious about the way we handle them in the clinical care situation, starting from the Tuskegee experiment and since. But I think there's a lot of fear of this vaccine, and the reason for that is that it's a deadly virus. I think people were afraid of the Ebola vaccine just because it's a deadly virus, and people assume if it's a deadly virus, like anthrax, for example, there were many in the military who were hesitant to get the anthrax vaccine, not that they had a choice, but people assume wrongly that if the disease is awful, like anthrax, Ebola, SARS-CoV-2, that therefore the vaccine is more likely to be dangerous, which is absolutely not a reasonable thing to think. So I think, again, this goes under managing expectations. We have to be really good about explaining this vaccine and what we can and can't expect from it.
Back to children, people are worried about multi-system inflammatory syndromes in children, right? Are there any possible long-term effects for children who develop the syndrome? What's your opinion about this new development?
Time will tell. I mean, it was called early on Kawasaki's-like disease. It's really not Kawasaki's. This particular syndrome affects an older group of children. Kawasaki's is more the 2 to 6 year old. This is more the older child, including young adults. So this, it's not Kawasaki. It's also the cytokine profile in these kids is different than the cytokine profile. And these kids have, for example, low platelet counts, whereas Kawasaki has high platelet counts. So it's a different disease. In terms of what are the long-term effects, we'll see. Certainly we know that it causes vasculitis of coronary arteries, which can be fatal. So in that way, it does mimic Kawasaki.
There has been talk that the virus is sensitive to heat. Is the increase in temperature affecting the virus?
Yeah, that's what you would have hoped, that increased temperature and increased humidity would affect the virus because it's an envelope virus. Typically, when you have viruses like these respiratory viruses, which are spread by small droplets, as the humidity gets higher, the small droplets accrete or acquire water more when in a humid climate, and therefore the droplets don't stay in the air as long. They drop more quickly, and therefore it's less contagious. But if you look at outbreaks in India now, outbreaks in Brazil, where the climates are warm and humid, it doesn't reassure you that we're going to have a dramatic drop. And we are already in June, and still, we saw, I think, 1,000 deaths in this country yesterday. So I'm not getting this feeling, we don't see any flu in this country now. Or the flu that we see is trivial. But yet we see this virus still circulating. So it's not flu, even though they're both envelope viruses. This is different.
So since you mentioned flu, I had a question for you. Historically, how do you compare this pandemic to previous disease outbreaks? You mentioned a couple, but how would you compare it with pandemic flu?
Yeah, I was wrong about this virus. I think I was on CNN International with Christiane Amanpour, and this was back at the beginning of March. I think it was after we experienced our first death in this country. And I predicted that this wouldn't be flu. It wouldn't be as bad as flu. I was dead wrong. So what did flu do this year in the United States? Flu this year affected about 30 million people. That's about a tenth of the population. It caused 700,000 people to be hospitalized and 62,000 people to die. Then, when we had our first death, could not imagine that this would be as bad as our routine epidemic flu every year. But I was wrong. This is clearly worse. And it also extends seems to be extending into the warmer time. So, flu, you can count on flu doing what it did last year, next year and the year after. The thing we have to worry about now is that assuming that this virus, SARS-CoV-2, is back again with us at a greater force in the winter, because typically respiratory viruses like this are winter viruses, that it's going to be now occurring at the same time as influenza. Here, influenza was starting to come down a little bit by March, but here, we could have experienced both at the same time, which would make you much more likely to overwhelm the healthcare system. We did a pretty good job at flattening the curve and not overwhelming the healthcare system, but I think next year will be tougher, making it all the more important to get an influenza vaccine. The good news is that some of the companies that are making the SARS-CoV-2 vaccines also make influenza vaccines, so it's good to know that they are still committed to making all the influenza vaccine that they typically make.
One of our attendees is asking you to please discuss the challenge in getting the new vaccine to market, supply chain, need for refrigeration. What's your opinion about a quick distribution?
No, that's a great point. It gets you back to flu. Flu is recommended for everybody in this country over 6 months of age, and so it's hard to do that just in the medical home, i.e., the doctor's office. And so pharmacies, give flu vaccine. And occasionally schools give flu vaccines. Are we going to be willing to do that here too? I do think we will have tens of millions of doses to hundreds of millions of doses ready by the time that phase 3 trials are finished, assuming we do phase 3 trials for these vaccines, big ones, big phase 3 trials. But I think we will be able to have, when we have the sort of phase 1 group, which is people who are most at risk, I do think we'll have enough vaccine for that. And I think in all likelihood, these will be 2-dose vaccines. The only thing I just want to warn you about is that if you look at the BARDA vaccines, they're genetic vaccines. They're messenger RNA, vaccines, or these, replication defective, either simian or human adenoviruses. Those are the ones that went to the top of the list, is because they're the fastest to make. But just because you're fastest doesn't mean you're best, and we need to make sure we know that.
I have another question from our audience. You might have talk about that, but I will address the question. Will the phase 3 trials include children? How will we evaluate risk of vaccine causing the MIS-C?
Right. Again, I think these will be it's hard to know what caused this multi-system disease. I mean, we need to know what factors in the virus had precipitated that. That'll make us smarter about the vaccine. So you're right, we're sort of going in a little blind. I can tell you that the children are not, as I understand it, the way I'm on this NIH active group, they're not going to be the ones who are initially studied here. Now, they will be studied in Europe, I think, and elsewhere. So I think we will get some information. And we do ultimately want to vaccinate children because I think it'll make us feel better about children going back to school. But I think we're not going to know that initially. Again, we're going to learn as we go, which is always true. Just pick any medical breakthrough, and you will find a human knowledge. Nature gives itself up slowly, grudgingly, and invariably with a human cost. And I think there will be some human cost. We learn as we go. Always true. So hey, that's where I'm back to managing expectations.
Yeah. One of our board members is asking, what is the best answer when we ask how long are people infection, with mixed messages on symptomatic and asymptomatic shedding of this virus?
Yeah, I think the World Health Organization did not help us recently when they said that you can have asymptomatic shedding. What they should have said is pre-symptomatic shedding. I mean, which is true for every virus, every respiratory virus. I mean, you're most contagious with measles right before you develop symptoms, because the virus attaches, enters, reproduces itself in the nose and throat. Then you have viremia, it spreads to the lungs, spreads to the brain, spreads to the skin. That's when you're shedding the most virus. And then you develop symptoms. Symptoms is based on your immune response. It's your immune response that's associated with symptoms. And so that's when viral replication starts to decrease. So they weren't telling us anything new. They just said it badly. They shouldn't have said asymptomatic. They should have said pre-symptomatic. Now, functionally, that doesn't matter because you don't know whether somebody's about to develop symptoms or not. So therefore, everybody should be scared of everybody. Hence the need to wear masks. And that's the best way to stop that. But see, what's a little tricky here is that we're not testing. When we test for PCR, with either nary swabs or nasopharyngeal swabs, we're not testing for infectious virus. We're testing for viral genome. That's not the same thing. I mean, for rotavirus, for example, where I spent most of my career, when you're infected with rotavirus, which is an intestinal infection, you'll shed infectious virus in your stool for about 6 or 7 days. You'll be PCR positive for 6 months, even though you're not contagious. So you have to be careful with that. What's interesting about this virus, again, under the category of we learn as we go here, this is a single-stranded RNA virus, but it's messenger RNA. It's like rubella, which is also a messenger RNA virus. When you're detecting by PCR is messenger RNA, which is really short-lived. I mean, it's very labile messenger RNA. That's why the messenger RNA, nobody's worried about messenger RNA vaccine being unsafe because it's so short-lived in the body. What I worry about is that it's also short-lived outside the body, which is why it's going to need a fairly complex lipid delivery system, which itself may be problematic because lipids can be fever-inducing. So, I mean, we'll see. But so what's weird about this virus is you can be PCR positive for a long period of time, for weeks, and without clearly making infectious virus, which is odd, which means that what the virus is doing is it's continuing to make messenger RNA without making whole virus. That's an odd thing to do. But again, it's a bat coronavirus that we're just learning about.
Thank you. Of the 3 fastest track paths to vaccine, which is most likely to be the safest? What do you think?
Beats me. We'll learn when we do the phase 3 trials.
What do you think about the origin of the virus, of this virus?
Well, I think it's the same thing as the origin of MERS and SARS. MERS and SARS, which is to say that it was a recombination event that occurred in vivo, probably involving a bat and a pangolin, which I'd never heard of actually until this virus emerged. That was the origin. I mean, this sort of notion that it was a lab accident or that it was an attempt to actually a germ warfare was just utter nonsense. It's just the same thing as SARS and MERS. It's an in vivo reassorted event, and it won't be the last one.
Do you think this pandemic has made people more or less receptive to getting vaccinated?
Hard to know. I hear it both ways. On the one hand, people really want a vaccine. On the other hand, they're a little scared to get it, which is actually, oddly enough, was also true of the polio vaccine. I mean, people were like just happy that there was a vaccine. Uptake was fairly slow. Now, part of it was the Cutter incident, which one of the companies that made that polio vaccine at warp speed back then, because we were making it without knowing whether the phase 3 trial would show that the virus was safe and effective. One of the companies, 5 companies made that vaccine: Eli Lilly, Park Davis, Pittman Moore, Wyeth, and Cutter Laboratories of Berkeley, California. It was the last vaccine Cutter Laboratories ever made because they made it badly. What they did was they failed to inactivate poliovirus in 2 of their lots. And as a consequence, about 120,000 children in 1955 were inadvertently inoculated with live, fully virulent poliovirus. About 40,000 developed abortive or short-lived polio. 164 children were permanently paralyzed and 10 were killed. I think it was the worst biological disaster in this country's history. And it wasn't, people try and blame that on the fact that these vaccines were made at risk. But that really wasn't it. I think we were just, as always, learning as we went. It was a scale-up problem. Wyeth also made a vaccine that paralyzed and killed children. We just had to learn, and we did. Very quickly, we had better safety tests. Very quickly, we had better ways of mass producing that vaccine within a matter of months. But again, there was a human price to pay for that knowledge.
Thank you. As you might know, we do run the Essex Metro Immunization Coalition, and we've seen reduced access to immunization for kids. And our question is, how do we communicate the risk of future vaccine preventable disease outbreaks like measles, pertussis, due to reduced vaccination coverage rates during this pandemic?
Yeah, it's worrisome, isn't it? I mean, we now have an under-vaccinated childhood population because we haven't been getting measles vaccine and haven't been getting pertussis vaccine, which are certainly still circulating in the United States. Pneumococcus still circulating in the United States. So you do worry about that. I think it'll be interesting to see how this plays out because, when we started to shelter in place in, although late, when we finally did start to do that, we were still in the flu season. And there was a fairly dramatic drop in flu associated with sheltering in place. Here, we still may be sheltering in place by the time we hit the winter again. And so that'll decrease, actually, the spread of measles or decrease the spread of flu. So that sort of works for you. But we'll see, obviously, what works against you is you have an under-vaccinated population. It's a shame, actually. I know at Children's Hospital of Philadelphia, what we had asked our chaplain practices to do was to bring in everybody less than 2. Because really, you really do need your priming series at 2, 4, and 6 months of age. Weigh them, measure them, give them their vaccines, send them home. Just do that, and then anything else that you needed to do, like anticipatory guidance, you could do by phone. But it really wasn't the doctors who weren't compliant with that. I think the doctors wanted to be compliant with that. I don't know how it works with your groups, but it was the parents. They just didn't want to go, to a place where other children may have SARS-CoV-2, so they didn't go. So yeah, we're going to be dealing with under-vaccinated childhood population heading into the winter.
Yeah, and speaking about that, what is your projection with reopening of schools, especially in New York, New Jersey area where the virus had hit hard? Should school reopen? If so, what would school look like to offer protection to students and staff?
Yeah, so I'm probably not the best person to answer that question. David Rubin, who's head of policy lab at Children's Hospital of Philadelphia, he's come out to say he thinks schools should reopen. Elementary school, junior high, high school, colleges. This is generally not an at-risk group, and there are downsides to not reopening, not the least of which is children staying home, parents not being able to go to work and hanging out with grandma and grandpa. So I do think the benefits actually of reopening are greater than the risks, which I think are small. So I personally think that we should reopen schools come September.
That's reassuring, thank you. What do you think is driving the unwillingness of some of the people to follow up guidelines like social distancing, wearing masks? How can we address the necessary behavior change and knowledge gaps? And how do you protect yourself during this time?
Yeah, we obviously are going to look back on this and realize there were things we should have done dramatically differently. We should have shut down entrance from Europe and China much earlier than we did. We should have done much more extensive testing in a manner like South Korea. Now, South Korea is not the United States. I mean, the culture is very different, and they were able to do a lot of things there they could do that we would have trouble doing here. But we should have been doing 5 million tests a day. We should have been doing at least 30 million tests a week. We weren't. I mean, we have a president who stands up now and talks proudly about how he's doing 2.5 million tests a day. That's still way. I'm sorry, 2.5 million tests a week, which is way less than we should be doing. So that's number one. We should, in terms of, I'm sorry, Liliana, tell me the original question again. Now that I've veered off course.
I think someone would like to know, how do you protect yourself, Dr. Offit?
how do I protect myself? I wear a mask when I go outside. I mean, so if we're not going to do testing, I think testing is now a lost phenomenon. We're not going to do it. We haven't been doing it. We're not going to do it. We haven't been doing the contact tracing we should do. So at the very least, wear a mask, wash your hands, social distance. That's the best thing you can do to protect yourself. It's just really frustrating that we aren't really good at that. And again, we haven't had leadership. I'm not just talking about the federal level, also at the state level. You have, this is somehow for whatever reason has become a political thing. Like the Republicans are less likely to wear a mask than the Democrats. I don't honestly do not understand how we came to that, but that's the least you can do to protect, not only yourself, but to protect others from you. It's selfish not to wear a mask.
It's a responsibility of all of us. I agree. How can we successfully educate the public about asymptomatic transmission of COVID-19 and other diseases and the need for vaccination?
Right. Again, I think people, it's what makes this virus so frightening is that there probably is a virus that lives on surface that you can pick up by touching a surface. The CDC sort of gone back and forth on this, but washing your hands is probably a good idea. And secondly, people can transmit the virus asymptomatically, so actually more likely pre-symptomatically. If you're asymptomatic, you're probably not going to go on to develop coughing and sneezing by definition, and so you're less likely to transmit that way. But so that's pretty frightening. Therefore, everybody you come in contact with, or arguably anything you touch, is potentially a way to get this virus. So it's frightening. Normally, I mean, if you're during the days of smallpox, and smallpox had a mortality rate of 30%. During the days of smallpox, you got infected if you were within 5 feet of someone who had smallpox. It's not small droplet spread. It's large droplet spread. But you knew who had smallpox. You knew because they had pox lesions on their face. And the reason that you got smallpox is that those blisters were also on the inside of their mouths. They ruptured, and then it was spread by large droplet. But if you were sitting 2 rows in front of an airplane, 2 rows in front of somebody with smallpox, you weren't going to get smallpox. But here, it's like if you're within 6 feet of anybody who's asymptomatic, you can get it. And so therefore it's frightening, but it's fatiguing. It is, I think we are under pandemic fatigue. And so people are just tired of this. They're tired. First of all, wearing a mask is not fun. And how do you handle all this? I'm on the board of trustees at the Franklin Institute, which is a science institute in Philadelphia. They're trying to figure out how to reopen. And so they're doing everything they can. They're asking people to wear masks. If you don't have a mask, one will be provided for you. They're asking children to wear masks, which is really hard because if you ever watched a young child wear a mask, it's like the opposite of wearing a mask, the way they dig around in that mask. But they're trying to have the degree that you can have social distancing in a museum. But the fact of the matter remains that the minute you walk outside, when this virus is still circulating, you're taking a risk. You are. There is no risk-free position, no matter how good you are about social distancing or wearing masks. There's still a certain risk. If you want to avoid SARS-CoV-2, if you want to avoid being infected with this virus, stay inside for the next couple of years. Stay inside, because the minute you walk outside, you are taking a risk. I mean, it is hard to watch because I'm on the board of the Franklin Institute to watch what they're trying to do, because they know they're going to get sued. They are. SEPTA has already been sued in Philadelphia for someone who got the virus while being on the bus, although I don't know how you prove that. But because they know they're going to get sued. And the truth is, this happens every year with influenza. 62,000 people died of influenza this year. I mean, it's not 110,000 or 115,000, which is where we are with this virus, but that's still a lot of people. But we take that risk every year. We're willing to take that risk. We're unwilling to take this one. But the fact of the matter is, we are taking that risk by walking outside and going to museums and eventually going to school, et cetera, that you can't eliminate the risk. All you can do is mitigate the risk the best that you can. And we just have trouble with that. Certainly personal injury lawyers seem to have trouble with that because they believe in absolute safety, which of course is a concept that doesn't exist.
Yeah, and there should be a balance. In New Jersey, we are going to have daycares open on the 15th. And as you mentioned, children will be challenged to wear a mask, and actually under 2 years old, they are not recommended for young children. So it is going to be a risk that many families and businesses will take. I have another question about the disease, the COVID-19. Why is it that some people, healthy people, have such mild symptoms or no symptoms at all, while others are developing such severe complication with COVID-19? And are there any other examples of diseases that follow similar paths?
I don't know. I mean, I'll give the answer that infectious disease people give when they get asked questions like this, you know, a unique interaction between the virus and the host. You know, we're an outbred population. Some people are more likely to be severely affected by a virus than others, presumably based on their major histocompatibility complex. So I don't know. Hopefully we will learn that over time.
Yeah, a lot of parents ask for their children to be tested for COVID antibodies. What do you think about that?
I don't think it's helpful for a couple of reasons. One, people who get an antibody test, and it's going to be a binding antibody test. It's not going to be a neutralizing antibody test. So it doesn't really tell you whether or not you're protected. So I think people who get a test and find that it's positive may falsely believe that they're protected, and therefore be more likely to not wear a mask or not engage in social distancing, et cetera. So I don't think it's helpful, actually. It doesn't tell you whether you're protected, which you're hoping it tells you.
I have another question from a board member. What is your opinion about universal testing for pregnant women? This has been started at many hospitals in New Jersey.
I mean, the good news is pregnant women don't seem to be at high risk, nor their babies here. So that's good. Again, you know, which is not true of flu. Flu, for example, you're 7 times more likely to develop pneumonia and be hospitalized with flu if you're a pregnant person than if you're a person of the same age who's not pregnant. So that's not true with this virus. This virus apparently doesn't seem to cause people to be more likely to hospitalize if you're pregnant than not pregnant. So that's good news. So the testing therefore becomes less important, I guess, to me.
Thank you. In New Jersey, the pools are going to open, and people are worried about, you know, safety to be outside in a pool with the virus. What do you think about transmission through water or through pools?
No, I don't think this is a waterborne transmission, but I mean, I think, you know, there are advantages to being outside. You are less likely to be infected if you're outside than inside, just in general. But again, I mean, you know, we're doing a grand national experiment. We never had a clear directive from the federal government or even some state governments about what to do. And so we're just, you know, some people are going out, some people aren't, some people are wearing masks, some people aren't. You look at a scene in Las Vegas at one of the casinos, nobody had a mask on. They were all as close together as possible. And we'll learn in a couple, 2-3 weeks exactly what it meant to do all that. We learned.
Yeah, one more. I think we have time for a couple of questions only. One more question about pregnancy, even though that's not your specialty, but the question is, do you think states or CDC should track SARS-CoV-2 in pregnancy to gain more information about perinatal transmission and increase knowledge about documented placenta thrombosis? Sure.
I think again, I think this virus only really started at the end of last year. We're only in the middle of this year. This is a novel virus. We're learning. Sure. I think gathering information about potential high-risk groups is always a good idea.
And one last question. Do you anticipate a less severe flu season if people continue to social distance and practice good hand hygiene, mask wearing in public? And if so, how do we increase flu vaccine uptake in the fall if people feel as though they are already protected from catching respiratory illness?
Yeah, I think you know, the flu virus, influenza, killed 62,000 people this year. We could have avoided that by staying inside. If every year, if we shelter in place in the winter, we will have far fewer flu deaths. So I think we'll have lesser. We did have lesser flu at the end of last year. Sorry to come down a little more precipitously. You can't do that. That's true with this virus too. I mean, the price we're paying is the second part of this public health nightmare. I mean, the first part was suffering and hospitalization and death caused by the virus. The second part of this public health nightmare is massive joblessness, which will, you know, no doubt lead to the kinds of things that you see with massive joblessness, which is food insecurity, homelessness, increase in domestic violence, which has already happened, increase in child abuse, which has already happened, increase in mental health issues such as depression and suicide, which has already happened. I mean, I think, you know, this is part two, which also has to be considered. And so you have to balance those two things, realizing that there's no risk-free situation. And we'll see as we move forward. But I do think we should do the best we can to try and get back to work. So wear masks at least, and get back to work in those situations where you can at least have some social distance.
Yeah, and that's true. This public health crisis will be amplified by many other crises, as you mentioned, economic and any other, public health issues. Thank you so much, Dr. Offit.